Hello there, and welcome to the Sleepy Facts channel. I'm so glad you found your way here. Maybe you had a long day, or maybe you're the kind of person who likes falling asleep to the gentle rhythm of waves rather than counting sheep. Either way, tonight's journey is for you. Let your mind relax, your breathing slow, and your body grow light as if floating in the shallows where land meets sea. Because tonight, we're exploring tide pools, those small worlds caught between ocean and shore. Filled with creatures that live by the rhythm of tides, we'll drift from the twice-daily transformation of these pools to the remarkable animals that call them home, discovering resilience in the smallest spaces. Some of what you hear tonight may surprise you, or at least spark curiosity as you drift off. Before we begin, if this helps you unwind, consider liking and subscribing, one sleepy soul at a time. Now, let's begin. Tide pools are small pockets of ocean left behind when the tide retreats. Temporary worlds that appear and disappear with the moon's pull on Earth's waters. Twice each day, the ocean rises and floods the rocky shore, and twice each day, it withdraws, leaving behind these isolated pools in the depressions and hollows of coastal rock. Each pool becomes a miniature ocean, a few feet across or perhaps just inches, holding within its boundaries a complete ecosystem that must survive until the sea returns. The pools shimmer in sunlight, mirror the sky, reflect clouds passing overhead. They are windows into the ocean, small enough to crouch beside and observe, vast enough to contain mysteries. These pools form along rocky coastlines, where waves have carved the stone into uneven surfaces over thousands of years. Water remains trapped in low spots, crevices, and bowls when the tide falls. The pools exist in the intertidal zone, that narrow band of shore that spends part of each day underwater and part exposed to air. This zone is one of the most challenging environments on Earth, subjected to dramatic changes in temperature, salinity, oxygen levels, and exposure to sun and wind. Yet life thrives here, adapted to conditions that would kill most marine creatures within hours. The organisms that live in tide pools are specialists in survival, masters of endurance. The twice-daily rhythm of tides follows the moon's gravity as it circles Earth. When the moon passes overhead, its gravitational pull draws ocean water toward it, creating a bulge of high water that we experience as high tide. As Earth rotates and the moon's position shifts, that bulge moves, and water level drops where you stand, revealing the intertidal zone. Because Earth rotates beneath the moon, most coastlines experience 2 high tides and 2 low tides each day, roughly 12 hours apart. This rhythm has persisted for billions of years, older than most life on land, and the creatures of tide pools have evolved to live by this ancient metronome. This leads to something even more remarkable about the variation in tides throughout each month. During new moons and full moons, when the sun and moon align, their gravitational forces combine to create especially high and especially low tides, called spring tides. During quarter moons, when sun and moon pull at right angles to each other, the tidal range is smaller, creating neap tides. Tide pool creatures experience these variations as expanding and contracting stress. Spring low tides expose them longer and to harsher conditions, while neap tides offer gentler transitions. The animals and plants must be prepared for both extremes, flexible enough to handle whatever the moon brings. Beyond the moon's influence, local geography shapes each tide pool's character. The size and depth of the pool, how sheltered it is from waves and wind, whether it sits high on the shore or low, where waves still reach it during low tide, all these factors determine what can live there. High pools may be isolated from the ocean for 8 or 10 hours during low tide, becoming warm in summer sun or chilled by winter air. Low pools reconnect with the sea more quickly, experiencing less dramatic changes. Each position in the intertidal zone presents different challenges, and different communities of organisms have adapted to each level. While the pools themselves are shaped by geology and tides, the creatures within them, transform these simple depressions into complex habitats. Barnacles cement themselves to rocks, creating rough surfaces where algae can cling. Mussels cluster together, their shells providing shelter for smaller animals. Seaweeds anchor to the bottom, their fronds creating forests in miniature, where tiny creatures hide from predators. Each organism modifies the pool, and these modifications create opportunities for other species. A tide pool is not just a container filled with seawater, but a community, each member affecting and being affected by all the others. Among the most visible residents of tide pools are sea anemones, creatures that look like flowers but are actually predatory animals. They attach to rocks with a muscular foot, remaining in one place for months or even years. Their bodies are cylindrical columns topped with rings of tentacles that wave gently in the current, each tentacle armed with thousands of stinging cells called cnidocytes. These cells contain coiled threads that fire when touched, injecting toxins and sticky substances into prey. Small fish, shrimp, or anything that brushes the tentacles becomes trapped and paralyzed, then drawn toward the central mouth where digestion begins. Despite their predatory nature, anemones move with such grace and stillness that they seem peaceful, gentle, swaying forms in the pool's gentle movement. The colors of sea anemones range from browns and greens to brilliant reds, purples, and oranges, depending on species and the algae they sometimes harbor in their tissues. Some anemones contain zooxanthellae, the same photosynthetic algae that live in coral, and these anemones gain nutrition from both hunting and the sugars their algae partners produce. The algae provide color and energy. The anemone provides a protected place to live and access to sunlight. It is cooperation at the cellular level, two very different organisms merged into what appears to be a single creature. When the tide is out and anemones are exposed to air, they pull their tentacles inward and close up, becoming smooth bumps on the rock, conserving moisture until the sea returns. Not all anemones remain in one spot throughout their lives. Some species can slowly glide across rocks using their muscular foot, moving just centimeters per day in search of better locations. Others can detach entirely and tumble in currents until they find a new place to settle. A few species can even swim in an awkward tumbling motion when threatened, pulsing their bodies to push through water. These movements are slow and deliberate, the anemone evaluating its environment and responding over hours or days rather than seconds. Time operates differently for anemones, each decision unfolding with patience that reflects their sessile lifestyle. Beyond this, there's another mystery in how some anemones clone themselves. Rather than reproducing sexually, they can split down the middle, one individual becoming two genetically identical anemones. This process, called fission, allows successful individuals to multiply and colonize good habitat quickly. A single anemone finding an ideal spot might split repeatedly, creating a cluster of clones all descended from that first settler. These clonal groups can dominate sections of tide pools, covering the rocks with identical copies of the original founder. It is a form of immortality through division, the original individual persisting in fragments that continue living long after the split. While anemones anchor to rocks, sea stars move across tide pool surfaces with purposeful slowness, traveling on hundreds of tiny tube feet that extend from grooves on their undersides. Each tube foot is a small hydraulic structure filled with fluid, and the sea star extends or retracts these feet through precise control of internal pressure. The feet have suction cups at their tips that grip rock surfaces, and by coordinating hundreds of these tiny grips, the sea star pulls itself forward in any direction, flowing over obstacles like living liquid. The movement is almost imperceptible unless you watch for several minutes, but the sea star is always going somewhere, always searching, always hunting in its unhurried way. Sea stars are predators. Despite their lack of obvious weapons or speed, they prey primarily on mussels, clams, and other shellfish, animals protected by hard shells that would seem impossible for a soft-bodied creature to penetrate. The sea star's solution is patience and persistence. It positions itself over a mussel, attaches its tube feet to both halves of the shell and begins pulling. The muscles hold its shell closed, but they must maintain constant tension, while the sea star can pull in shifts, some tube feet relaxing while others continue applying force. This tug-of-war can last hours, the sea star never giving up, the muscle gradually weakening. Eventually, the shell opens just a tiny crack, barely visible, but enough for the sea star's purposes. Through that small gap, the sea star everts its stomach, pushing the soft tissue out through its mouth and into the mussel's shell. The stomach surrounds the mussel's soft body and begins digesting it while still inside the shell, breaking down tissues with enzymes and absorbing the nutrients. This is external digestion, the sea star essentially eating its prey from the inside out without ever fully opening the shell. When the meal is complete, the stomach retracts back into the sea star's body, leaving behind an empty shell that will eventually open and fall away. It is hunting as slow siege. Victory through endurance rather than speed or strength. This hunting behavior shapes entire tide pool communities. Sea stars are keystone predators, keeping mussel populations in check and preventing them from monopolizing space on the rocks. Without sea stars, mussels would cover all available surfaces, crowding out other species. With sea stars present, mussels are controlled, creating space for barnacles, algae, and other organisms. The sea star maintains diversity not through any conscious intention but simply by eating its appetite, creating opportunities for species that could not compete with unchecked mussels. A single predator becomes an architect of community structure, its feeding habits rippling through the ecosystem. Not all sea stars hunt mussels. Different species specialize in different prey. Some eat barnacles, using their tube feet to pry individuals off rocks. Others consume algae, scraping it from surfaces with specialized feeding structures. Sunflower stars, the largest and fastest of all sea stars, hunt sea urchins, crabs, and even other sea stars. Each species fills a particular role, removing certain prey items and creating patterns of abundance and absence across tide pools. The diversity of sea stars reflects the diversity of available food. Evolution, discovering multiple ways to exploit the resources trapped in these small pools. While sea stars crawl across rocks, sea urchins move through tide pools using spines that double as walking legs. The urchin plants its spines against the substrate and levers itself forward, rotating to move in any direction. Progress is slow but steady, the urchin flowing like a spiny ball across the bottom. Those spines also provide protection, making the urchin difficult for most predators to handle or swallow. Sea otters have learned to crack them open against rocks, and some sea stars can flip them over to access the softer undersides. But for most predators, the urchin's spines present an impenetrable barrier. The urchin carries its fortress with it, safe within its own architecture. Beneath the spines, the sea urchin's body is a complex organism built around pentaradial symmetry, 2005-part organization around a central axis. The skeleton is made of calcium carbonate plates fused together into a rigid test, covered in skin and spines. On the underside, 5 teeth protrude from the mouth in a complex jaw structure called Aristotle's lantern. These teeth scrape algae from rocks, grinding plant material that the urchin processes in its long digestive tract. The urchin is essentially a mobile eating machine, crawling from one algae patch to another, grazing continuously like underwater cattle. Its feeding creates patches of bare rock, interspersed with algae-covered areas, adding heterogeneity to the habitat. This grazing behavior becomes particularly important in kelp forests and tide pools, where urchins can overgraze if their populations grow too large. In some areas, urchin populations have exploded after sea otter populations declined, and these urchin barrens become desert-like expanses of bare rock with little algae or associated life. The urchins eat themselves into starvation, consuming all available food and then waiting in a kind of suspended animation for new algae to grow. They can survive for years in these impoverished conditions, immediately consuming any new growth before it establishes. Controlling urchin populations is essential for maintaining healthy tide pool ecosystems. Balance between grazers and plants that allows both to persist. Beyond urchins and sea stars, tide pools host hermit crabs that solve the vulnerability of soft abdomens by living in borrowed shells. Unlike true crabs with armored carapaces protecting their entire bodies, hermit crabs have soft, curved abdomens that must be tucked into empty snail shells for protection. The hermit crab backs into a shell, grips the shell's interior with modified rear legs, and carries this mobile home wherever it goes. As the crab grows, it must periodically find larger shells, abandoning the old one and quickly scrambling into the new one before predators can exploit its moment of vulnerability. Tide pools after high tide often contain empty shells, remnants of dead snails that become valuable real estate for hermit crabs. The search for shells creates competition among hermit crabs, with larger individuals sometimes evicting smaller ones from desirable shells. When a particularly good shell appears, multiple crabs may line up by size, each waiting to move up one step in the housing market. The largest crab takes the best shell, leaving its old shell for the next crab, which leaves its shell for the next. A cascade of moves like musical chairs where everyone wins. This organized behavior requires some recognition of shell size and quality, some ability to evaluate resources and make decisions. The hermit crab is not merely reacting to stimuli, but assessing options and choosing among them. Tiny intelligence applied to the problem of staying protected. While hermit crabs carry their homes, true crabs rely on speed and aggression for defense. Small shore crabs scuttle across tide pool rocks, moving sideways with surprising quickness when threatened. Their hard carapaces protect their backs. Their claws can deliver painful pinches to predators, and their ability to squeeze into narrow cracks makes them difficult to catch. Shore crabs are omnivores, eating algae, small animals, and detritus, serving as cleaners that recycle nutrients. They patrol tide pools like miniature maintenance workers, dismantling dead organisms and returning their components to the ecosystem. Without crabs and other scavengers, tide pools would fill with debris, nutrients locked in decaying matter rather than available for living things. These crabs demonstrate remarkable navigation abilities. Despite having relatively simple nervous systems, they learn the locations of shelter within their territories, remember tide schedules, and can find their way home after foraging trips. When removed from their tide pool and released elsewhere, shore crabs can sometimes navigate back to their home pools over considerable distances, suggesting they use multiple cues including sun position, landmarks, and possibly Earth's magnetic field. This navigational sophistication allows crabs to exploit resources across multiple pools while maintaining a home base where they know the safe hiding spots and best feeding areas. They are not wandering randomly but traveling with purpose through a landscape they understand. Not all tide pool crustaceans are as large or visible as crabs. Tiny amphipods, copepods, and isopods swarm in the pool water and hide among algae, creatures barely larger than grains of sand. These small crustaceans form the base of many tide pool food webs, consuming microscopic algae and detritus, and converting these resources into animal protein that larger predators can eat. A single tide pool might contain thousands of these tiny organisms, their populations fluctuating with seasons and conditions. They are nearly invisible to casual observers, but essential to ecosystem function, the workers that keep nutrients cycling and energy flowing from primary producers to predators. Among these tiny creatures, some have evolved to handle the extreme conditions of high tide pools in particularly elegant ways. Certain copepods can survive complete desiccation, entering a state of suspended animation when their pools dry out entirely. Their metabolism stops, their cells protect themselves from damage, and they wait as dormant cysts until water returns. When rain or the next high tide refills the pool, these copepods rehydrate and resume normal activity within hours, as if no time had passed. This ability to shut down and restart life processes allows them to inhabit pools that would be uninhabitable for organisms requiring continuous immersion. They are creatures that can pause existence, waiting through the harsh times for conditions to improve. This capacity for dormancy appears in various forms across many tide pool species. Barnacle larvae can delay settlement if conditions are poor, remaining in the plankton longer than typical. Some algae produce resistant spores that survive extended dry periods. Fish eggs laid in high pools may pause development until appropriate tides arrive. These strategies represent different solutions to the same fundamental problem: surviving in an environment that is sometimes hostile and unpredictable. The tide pool selects for flexibility, for organisms that can wait, endure, and recover, rather than requiring constant optimal conditions. While small crustaceans hide among algae, larger organisms shelter beneath overhangs and in crevices where conditions remain more stable. Blennies, small fish with elongated bodies and large eyes, wedge themselves into tight spaces during low tide, waiting for the ocean's return. These fish can breathe air for short periods, absorbing oxygen through moist skin when trapped in pools with low oxygen levels. They also tolerate extreme temperature fluctuations, surviving in water that might reach 30 degrees Celsius or more during summer low tides. Their adaptations allow them to occupy a niche that most fish cannot tolerate, making them among the most successful vertebrates in tide pool environments. Blennies exhibit complex behaviors despite their small size. Males establish territories around good egg-laying sites and perform courtship displays to attract females, bobbing their heads and flashing their fins. After mating, males guard the eggs, fanning them with their fins to ensure adequate oxygen and defending them from potential predators. This parental care continues until the eggs hatch and the larvae swim away into the plankton. The male blenny invests significant time and energy in his offspring's survival. Unusual behavior for fish, but advantageous in the contained environment of a tide pool, where securing good breeding sites and protecting young increases reproductive success. Not all fish in tide pools are permanent residents. Many species use pools as nurseries, larvae settling from the plankton and juveniles growing in the relatively protected pool environment before migrating to deeper water as adults. Tide pools become kindergartens for the ocean, safe places where young fish can learn to hunt and avoid predators before facing the open sea. The pools provide abundant food in the form of small invertebrates and algae, and the shallow water makes it difficult for large predators to enter. Young fish pay for this safety with exposure to temperature swings and the occasional pool that dries completely, but overall the trade-off favors survival. Many adult fish in offshore waters began their lives in these small pools. Beyond fish and crustaceans, tide pools host snails that graze on algae-covered rocks, moving at speeds that make sea stars look fast by comparison. Limpets and periwinkles rasp algae from surfaces using their radulas, specialized feeding structures covered in tiny teeth. They move methodically across the rocks, creating visible grazing trails in the algae film. Some species return to the same home scar after each foraging trip, a small depression in the rock that matches their shell perfectly and helps prevent water loss during low tide. The snail wanders perhaps a meter from home, feeds for hours, then follows its own mucus trail back to the exact spot where it started. It is navigation by chemistry, following the trail it laid during its outbound journey. These mucus trails serve multiple purposes beyond navigation. The mucus protects the snail's soft foot from rough rocks, reduces friction to make movement easier, and may contain chemical signals that other snails can detect. Some species appear to follow each other's trails, creating highways across the rocks where multiple individuals travel the same routes. The mucus eventually breaks down, recycled by bacteria, but while it remains, it influences the movement patterns of the snail community. A tide pool's rocks are covered in invisible pathways, a transportation network written in chemicals that slowly fade and must be constantly renewed. While snails graze on algae, Other mollusks filter food from the water itself. Mussels cluster together in dense beds, each individual anchored to rocks by tough threads, called byssal threads, that the mussel produces from a gland in its foot. These threads are remarkably strong, composed of proteins that combine flexibility with tensile strength, allowing mussels to withstand wave forces that would tear most attachments apart. The mussel secretes liquid proteins that harden on contact with seawater, building its anchor thread by thread until it is firmly attached. Once established, the mussel opens its shell slightly and pumps water through its body, filtering out microscopic algae and organic particles for food. This filter-feeding lifestyle makes mussels crucial components of tide pool ecosystems. A single mussel can filter several liters of water per hour, removing suspended particles and clarifying the water. A bed of hundreds or thousands of mussels can process all the water in a tide pool multiple times each tidal cycle, dramatically affecting water quality and nutrient availability. The mussels concentrate nutrients from the water into their bodies, making those nutrients available to predators like sea stars. They also produce waste that settles to the bottom, enriching sediments and feeding detritus-based food webs. The mussel bed becomes a living filter system, maintained by organisms pursuing their own survival but collectively transforming the environment. These mussel beds also provide habitat for other species. Small crabs, worms, and amphipods live among the mussels, hiding between shells and feeding on food particles the mussels drop or on the algae that grows on mussel shells. Young sea stars often settle in mussel beds, finding both food and shelter among the clustered shells. Some species of fish lay their eggs among mussels, using the complex structure as protection for developing young. The mussel bed is not just a cluster of mollusks but an entire community. The mussels creating three-dimensional habitat that increases the diversity and abundance of life in the pool. Each mussel contributes to a structure larger than itself, and that structure supports life beyond the mussel's own. Beyond the animals, tide pools contain diverse algae that form the foundation of most tide pool food webs. These photosynthetic organisms range from single-celled diatoms too small to see individually to large brown kelps several feet long. The algae coat rocks in films, form bushy tufts, drape from overhangs like curtains, and carpet pool bottoms in meadows of green and red. During low tide, when sun shines directly into pools, these algae photosynthesize intensely, producing oxygen that bubbles from their surfaces and raises oxygen levels in the water. This oxygen production sustains the animals in the pool, providing the dissolved gas they need to breathe. The colors of tide pool algae reflect different pigments adapted to different light conditions. Green algae contain chlorophyll similar to land plants, thriving in bright, shallow pools. Red algae contain additional pigments that allow them to photosynthesize in deeper pools or shaded areas where less light penetrates. Brown algae, including kelps, dominate mid-level pools where they can tolerate moderate wave action. Each type of algae has found its niche within the intertidal zone, sorted by tolerance for exposure, wave force, and light availability. The algae create a vertical zonation pattern, different species dominating at different heights on the shore, painting bands of color across the rocky intertidal that shift with the tides. These algae must endure remarkable challenges during low tide. Exposed to air, they face desiccation as water evaporates from their surfaces. Summer sun can heat exposed algae to temperatures far above what they experience underwater, and on hot days the tissue can begin to cook, proteins denaturing if temperatures climb too high. Winter brings different challenges. Freezing air temperatures, ice forming on fronds, cells damaged by ice crystal formation. Yet tide pool algae survive these extremes through various adaptations. Some produce mucilage coatings that retain moisture during exposure. Others can tolerate losing most of their water content, shriveling during low tide but rehydrating when the sea returns. Many contain compounds that protect against ultraviolet radiation, natural sunscreens that prevent damage from intense sun exposure. This resilience allows algae to colonize the highest reaches of the intertidal zone, places that spend more time exposed to air than submerged in water. These high-zone algae are the pioneers, the extremophiles of the shore, surviving conditions that would kill most marine organisms. They grow slowly, reproduce whenever possible, and persist through patience rather than rapid growth. Some species can survive weeks without submersion, waiting for spring tides or storm waves to bring relief. They are living proof that the boundary between marine and terrestrial life is not sharp but gradual, a transition zone where organisms adapted to water slowly give way to organisms adapted to air. While algae provide food through photosynthesis, other organisms recycle nutrients by consuming detritus, the fragments of dead organic matter that accumulate in tide pools. Tiny worms burrow through sediments at pool bottoms, processing organic particles and aerating the substrate. Sea cucumbers extend feeding tentacles that collect settled detritus and transfer it to their mouths. Brittle stars, relatives of sea stars, emerge from hiding places at night to feed on whatever organic matter they can find, their long, thin arms sweeping across surfaces and gathering food particles. These detritivores form an essential link in nutrient cycles, breaking down complex organic molecules and making their components available for other organisms to use. The brittle stars themselves are remarkable creatures, similar to sea stars but with much longer, more flexible arms that they use for both locomotion and feeding. Each arm can move independently, the brittle star flowing across the bottom, like a living hand with 5 thin fingers. When threatened, brittle stars can detach arms as a defense mechanism, leaving the writhing arm to distract predators while the main body escapes. The lost arm eventually regenerates, growing back over weeks or months until the brittle star is complete again. This ability to sacrifice and regrow body parts represents an extreme adaptation to predation, pressure, trading temporary loss for survival. The brittle star treats its own limbs as expendable, prioritizing the central disc where vital organs reside. Not all brittle stars feed on detritus. Some are suspension feeders, raising their arms into the water and catching food particles that drift past. Others are predatory, using their arms to capture small crustaceans and worms. The diversity of feeding strategies within this single group of animals demonstrates how evolution explores possibilities, finding multiple ways to make a living within the same basic body plan. Each species of brittle star has specialized slightly differently, reducing competition with relatives while all maintaining the characteristic 5-armed form. They are variations on a theme: nature's experiments in what arms can accomplish. Beyond brittle stars, tide pools contain chitons, mollusks that look like small armored ovals clinging to rocks. The chiton's shell consists of 8 overlapping plates surrounded by a tough girdle, giving it flexibility to conform to uneven rock surfaces while maintaining protection from predators and wave forces. Chitons grip rocks with a muscular foot that creates suction, and their hold is so strong that trying to pry one off often damages the animal or breaks the rock before breaking the seal. They feed by scraping algae from surfaces with their radulars, moving slowly across territories that they know intimately, returning to favorite resting spots after each foraging session. These kittens demonstrate homing behavior similar to limpets, navigating back to specific locations on rocks where they fit perfectly against the surface. Some researchers believe kittens recognize their home spots by chemical cues, while others suggest they may use visual landmarks or simply remember the route through repeated travel. Whatever the mechanism, the result is impressive. A chiton displaced from its home can often find its way back across complex rock surfaces, choosing the correct path at each decision point. This navigational ability suggests more sophisticated cognition than typically attributed to such simple-looking creatures. The chiton's brain is small, but it contains enough processing power to solve the spatial problems necessary for survival. While chitons move slowly across exposed surfaces, other tide pool residents hide beneath rocks, where conditions remain more stable, and predators have difficulty reaching. Flipping a tide pool rock reveals an entirely different community. flatworms gliding across surfaces, ribbon worms coiled in crevices, sea spiders walking on improbably thin legs, tiny fish sheltering in the coolness beneath. These cryptic species avoid the challenges of exposure by choosing permanent shade, accepting reduced feeding opportunities in exchange for environmental stability. The underside of rocks becomes its own microhabitat, cooler and more humid than exposed surfaces, a refuge that supports species unable to tolerate full exposure. This hidden diversity reminds us that tide pools contain more life than meets the casual eye. For every organism visible from above, several more hide in crevices, beneath rocks, or within the sediment. Some emerge only at night when predation risk decreases. Others remain hidden throughout their lives, reproducing and dying without ever experiencing direct sunlight. The tide pool is layered, three-dimensional, filled with hiding places that create opportunities for specialists adapted to particular microhabitats. To truly know a tide pool requires patient observation over many visits, learning which species emerge at different times and which prefer different conditions. Among the night-emerging creatures, some octopuses shelter in tide pools during low tide, waiting for the ocean's return before hunting. These small octopuses are specialists in enduring exposure, able to survive in pools with elevated temperatures and reduced oxygen. They squeeze into the tiniest crevices, their boneless bodies compressing to fit spaces that seem impossibly small. During the day, they remain hidden and inactive, conserving energy and oxygen. When high tide returns and water quality improves, they emerge to hunt crabs and small fish, using intelligence honed over millions of years to solve the problem of catching prey in confined spaces. The octopus brings cognitive sophistication to environments that most intelligent animals avoid. This leads to something even more remarkable about how some tide pool organisms synchronize their activities with tidal rhythms. Many species have internal biological clocks that track the roughly 12-hour tidal cycle, allowing them to anticipate tide changes and prepare appropriately. Crabs become more active as high tide approaches, positioning themselves to feed when fresh water brings new food. Fish increase activity levels before submersion, preparing to hunt or mate. Some barnacles begin opening their shells slightly before high tide actually arrives, readying themselves to filter feed as soon as water quality improves. These internal rhythms persist even when animals are removed from tidal environments, suggesting that the timing mechanisms are genetic rather than learned, evolved over countless generations to match the moon's reliable schedule. The precision of these biological clocks fascinates researchers studying how organisms measure time. Some species appear to track not just the 12-hour tidal cycle but also the 14-day spring neap cycle and even annual patterns of seasonal change. These multiple overlapping rhythms allow animals to prepare for predictable changes in their environment, giving them competitive advantages over species that can only react after conditions have already shifted. The tide pool organism that anticipates tomorrow's conditions has more time to prepare than the one that waits until tomorrow arrives. Natural selection has thus favored the evolution of internal clocks, genetic timekeepers that track the cosmos and translate astronomical movements into behavioral programs. While some organisms track time, others track space, maintaining territories within tide pools and defending those territories against competitors. Some fish species establish and defend small areas containing good shelter and feeding spots, chasing away intruders of the same species. Male crabs may defend territories that include multiple females, preventing other males from mating opportunities. Even some sea anemones show territorial behavior, using special aggressive tentacles to sting neighboring anemones that grow too close. These territories are tiny by terrestrial standards, often just a few square centimeters, but they represent valuable resources in the crowded tide pool environment, where space is limited and competition is intense. The defense of territories requires recognition of boundaries and competitors, cognitive abilities that many tide pool organisms possess. A territorial fish must distinguish between harmless visitors and competitive threats, between males that challenge its dominance and females that represent mating opportunities. The fish must remember the boundaries of its territory and patrol those boundaries regularly. It must assess whether potential intruders are worth fighting, making cost-benefit calculations about when to attack and when to retreat. These decisions happen quickly, the fish processing information and choosing actions within seconds. Yet the underlying cognition is sophisticated, requiring memory, recognition, and strategic thinking. Not all interactions in tide pools are competitive. Many species cooperate or at least coexist peacefully, sharing space and resources without conflict. Small fish hide among sea anemone tentacles, protected by the stinging cells that would harm predators, but somehow do not harm the symbiotic fish. Cleaning shrimp set up stations where larger animals come to have parasites removed, an exchange of services that benefits both parties. Algae provide shelter for tiny invertebrates, which in turn may defend the algae from grazing herbivores. These positive interactions create interconnected webs of relationships, each species affecting others in complex ways that propagate through the community. Among the most important positive interactions are those between species and the physical structures they create. Mussel beds, as mentioned, provide habitat for many other species. Similarly, sea urchins create hollows in soft rock as they graze, and these hollows become shelters for small animals. Barnacle clusters create rough surfaces where algae and small invertebrates attach. Each organism that modifies its environment potentially creates opportunities for others, and over time, these accumulated modifications transform bare rock into complex habitat, supporting high biodiversity. The physical structure of a mature tide pool is not just geological but biological, shaped by the activities of countless organisms over years or decades. This biological structuring of habitat means that tide pools change over time, following predictable patterns of ecological succession. A newly created pool, or one scoured clean by winter storms, begins with bare rock. Pioneer species like algae and barnacles colonize first, settling from the plankton and establishing initial populations. These pioneers modify conditions, making the pool more suitable for later colonizers. Gradually, predators arrive and begin controlling prey populations. Longer-lived species, like anemones and mussels, become established. Eventually, the pool reaches a quasi-stable state where births roughly balance deaths, and the community composition becomes relatively predictable. But this equilibrium is always temporary, vulnerable to disturbance that can reset succession to earlier stages. Disturbances come in many forms. Winter storms can flip boulders, destroy mussel beds, and scour pools clean of life. Unusually hot weather can raise temperatures to lethal levels, killing sensitive species. Predators moving through can eliminate key species, cascading effects through the food web. Fresh water from heavy rains can dilute salinity beyond tolerance limits. Each disturbance creates opportunities for recolonization, resetting parts of the community to earlier successional stages. The tide pool exists in constant flux, stability, and change, balancing over time scales from days to decades. What you observe in a pool today may look quite different from the same pool a year ago or a year hence. This dynamic nature makes tide pools excellent natural laboratories for studying ecological processes. Researchers have used tide pools to investigate competition, predation, succession, and community assembly, often conducting experiments that would be impossible in more complex environments. The small size of pools makes them manageable study systems, and their isolation means experimental manipulations in one pool do not affect adjacent pools. Scientists can remove predators from some pools and compare them to unmanipulated controls, testing hypotheses about top-down control of food webs. They can add nutrients to some pools and observe the effects on algae and herbivores. Decades of such research have made tide pools among the best understood ecosystems on Earth, yet mysteries remain. We still do not fully understand how larvae settling from the plankton choose where to attach, what cues they use to identify suitable habitat. We know little about the role of microorganisms, bacteria, archaea, tiny fungi, in tide pool nutrient cycles. We are only beginning to appreciate how individual variation affects ecological outcomes, how personalities of individual sea stars or crabs might influence community structure, and we have barely begun to study how tide pools are changing in response to climate change, ocean acidification, and other human impacts. Each answer reveals new questions, and the humble tide pool continues, offering insights into how life organizes itself in challenging environments. Among the human impacts affecting tide pools, warming ocean temperatures are perhaps most pervasive. As sea surface temperatures increase, tide pools exposed during low tide absorb more heat from warmer air. Species already living near their thermal tolerance limits may be pushed beyond what they can survive. Some species are shifting their ranges poleward, tracking their preferred temperatures as climate zones migrate. Others are simply disappearing from pools where they once thrived, their former habitats now too warm during summer low tides. These shifts ripple through communities, changing interaction networks as species compositions change. The tide pool of the future may look quite different from the tide pool of the past. Ocean acidification presents another challenge. As atmospheric carbon dioxide dissolves in seawater, it forms carbonic acid, lowering the ocean's pH. This acidification makes it harder for organisms to build shells and skeletons from calcium carbonate, affecting mollusks, crustaceans, urchins, and many other tide pool species. Young barnacles may struggle to build their protective plates. Mussel shells may become thinner and more fragile. Coralline algae that cement themselves to rocks with calcium carbonate may weaken. The cascading effects could restructure entire communities, favoring species tolerant of low pH while eliminating those that depend on carbonate chemistry. We are running an unintended global experiment, changing the ocean's fundamental chemistry and watching how life responds. Beyond climate impacts, direct human disturbance affects many tide pools. Visitors walking across intertidal rocks can crush organisms underfoot or dislodge them from their attachments. Collecting creatures for aquariums or souvenirs depletes populations. Pollution from coastal development introduces toxins and excess nutrients. In heavily visited areas, the cumulative impact of many small disturbances can dramatically reduce biodiversity and alter community structure. Some popular tide pool areas have implemented protection measures, creating marine reserves where collection is prohibited and visitor access is managed. These protected areas demonstrate that tide pool communities can recover from human impacts when given the chance. Resilience built into these systems through millions of years of adapting to natural disturbances. This resilience comes from several sources. Many tide pool species produce abundant offspring, allowing populations to rebound quickly after die-offs. The constant influx of larvae from offshore populations can reseed tide pools, even if local populations are eliminated. The physical structure of the pools themselves persists through disturbances, providing template for community reassembly. And the species that live in tide pools are already adapted to extreme conditions, giving them tolerance that helps them weather additional stresses. The tide pool community is not fragile but rather surprisingly tough, capable of absorbing significant punishment while maintaining essential functions. Yet resilience has limits. Repeated disturbances can exhaust a community's ability to recover. Stresses that push species beyond physiological limits cannot be tolerated no matter how adapted the species. And some changes may permanently alter tide pool ecosystems in ways that prevent return to previous states. A pool heated to the point that all barnacles die may be recolonized by different species, better adapted to warmer conditions, creating a new community composition that persists even after temperatures moderate. Ecology speaks of alternative stable states, different configurations of species that can maintain themselves once established. Tide pools may shift between such states. crossing thresholds beyond which return to previous conditions becomes impossible. While we worry about human impacts, the organisms themselves continue their daily business, oblivious to the larger patterns we perceive. A limpet grazing on algae does not ponder climate change. An anemone catching plankton does not consider ocean acidification. Each organism simply pursues survival and reproduction, using the adaptations evolution has given it. If conditions become unsuitable, individuals die and populations decline. But this process is invisible to the organisms themselves. They do not grieve losses or celebrate successes. They merely persist or do not persist. There is something both pitiless and clean about this. Natural selection operating without emotion, sorting the adapted from the maladapted through the simple criterion of survival. This impersonal quality of natural processes makes tide pools excellent places for contemplation, for considering our own place in nature. When you crouch beside a tide pool and watch a sea star hunting a mussel, you witness interactions that have played out for millions of years. Relationships older than our species, older than most of the life forms we consider familiar. The limpet on the rock beside you is cousin to the limpets that watched dinosaurs pass, descendant of forms that survived mass extinctions that eliminated most life on Earth. These organisms connect us to deep time, to the vast sweep of evolutionary history that produced the living world. They make visceral the reality that we are newcomers to this planet, temporary participants in processes far older and larger than ourselves. Yet our newness does not diminish our impact. In the few thousand years of human civilization, we have changed the planet more dramatically than ice ages or volcanic eruptions. We have moved continents' worth of rock and soil, diverted rivers, filled the sky with our chemistry. We have caused extinction rates comparable to the great mass extinctions of the past. And we have done this not through malice, but through sheer numbers and technological power. The tide pool before you has likely been touched by human influence. Warmer water, more acidic, perhaps polluted, certainly more disturbed than tide pools experienced through most of their history. We cannot step outside our impact. We can only decide what kind of impact to have. The encouraging news is that conservation works when we commit to it. Protected tide pool areas show that damaged communities can recover. Reduced pollution allows sensitive species to return. Managing visitor access prevents overexploitation. These small successes do not solve all problems, but they demonstrate that negative trends can be reversed when we choose to act. The tide pool is resilient, and our impacts, while significant, need not be permanent. The same intelligence that allows us to damage ecosystems also allows us to protect them, to make informed choices about how we interact with the natural world. We can be mindful visitors rather than destructive intruders. Among the lessons tide pools teach is that small spaces can support remarkable diversity and complexity. A pool 10 feet across may contain dozens of species interacting in intricate food webs, competing for resources, cooperating in surprising ways, creating and responding to environmental change. Size does not determine ecological importance. Some of the most productive and biodiverse ecosystems on Earth are mangrove swamps, seagrass beds, coral reefs, habitats that seem minor compared to vast oceans or forests, but that support disproportionate amounts of life. The tide pool is kin to these systems, a small space that contains multitudes, proof that complexity can flourish in confined quarters when conditions permit. This complexity emerges from simple rules repeated across many organisms. Each species responds to local conditions: temperature, salinity, food availability, predation risk, TNT, according to its particular adaptations and tolerances. These individual responses aggregate into community patterns, and these patterns shift as conditions change. There is no master plan, no coordination among species to create balanced communities. The order you observe in a tide pool is emergent order, arising from countless small interactions among organisms pursuing their own survival. It is the same kind of order that creates all ecological systems, from tropical forests to deep-sea vents. Order without an orderer, pattern without a planner. This emergent complexity means that tide pools are never truly finished or complete. They are works in progress, communities in constant negotiation between stability and change. Each day brings new larvae, settling from the plankton, potential colonists that may establish or may fail. Each tide brings different conditions, different food availability, different predation pressure. Over seasons, temperatures shift, storm frequency changes, reproductive cycles of different species peak at different times. The tide pool responds to all these influences, its community composition adjusting continuously, finding temporary equilibria that persist until the next disturbance arrives. It is dynamic equilibrium, balance maintained through constant adjustment rather than fixed stability. Among the seasonal changes affecting tide pools, winter storms bring some of the most dramatic transformations. Large waves can restructure pools entirely, moving boulders that have been stable for years, stripping away established communities and creating blank surfaces for recolonization. Some organisms hunker down, retreating into protective crevices, or simply clinging tightly and enduring the battering. Others are torn away, their grip insufficient against the storm's power. After major storms, tide pools can look newly created, bare rock gleaming where moments before there was dense life. Yet within weeks, larvae begin settling, spores germinate, and the slow process of community assembly begins again. The pool recovers, demonstrating the resilience built into these systems through evolutionary time. Summer brings different challenges, particularly in pools at higher elevations in the intertidal zone. These pools may heat dramatically during daytime low tides, when direct sun beats down on confined water volumes. Temperatures can climb 20 degrees or more above normal sea temperature, creating thermal stress that tests the limits of every organism. Some species have enzymes that work across broad temperature ranges, maintaining metabolism despite heat. Others produce heat shock proteins that protect cellular structures from thermal damage. A few simply shut down, entering dormant states until temperatures moderate. The summer tide pool becomes a thermal gauntlet, and only species with appropriate adaptations can survive the challenge. These temperature extremes also affect dissolved oxygen levels, because warm water holds less oxygen than cold water. As temperatures climb, oxygen concentrations drop, and organisms must extract their needed gas from increasingly depleted water. Fish may become sluggish as they struggle to meet oxygen demand. Invertebrates may move to pool edges where wave splash provides slightly better oxygenation. Some algae photosynthesize more rapidly in warm conditions, producing oxygen that helps offset the reduced solubility. But this only helps during daylight hours. At night, all organisms respire, but no one photosynthesizes, and oxygen levels can drop dangerously low. The dawn following a hot summer night may find some tide pool organisms dead from oxygen depletion, victims of a perfect storm of high temperature and prolonged darkness. Beyond temperature and oxygen, salinity fluctuates dramatically in some pools. Heavy rain can dilute seawater, dropping salinity to levels that stress marine organisms adapted to ocean salt concentrations. Conversely, evaporation during hot weather can concentrate salts, raising salinity above normal. Both extremes challenge osmoregulation, the processes organisms use to maintain appropriate internal salt concentrations. Most tide pool species have evolved tolerance for salinity variation, able to adjust their internal chemistry to match changing external conditions. But this tolerance has limits, and extreme events, cloudbursts or weeks without rain, can create conditions that exceed even adapted species' abilities to cope. These fluctuations in temperature, oxygen, and salinity create a mosaic of conditions across different pools, even within small areas of coastline. A high pool exposed for 8 hours may experience very different conditions than a low pool exposed for only 2 hours. A deep pool with large water volume buffers temperature changes better than a shallow pool that heats quickly. A sheltered pool protected from wind and waves experiences different evaporation rates than an exposed pool. This environmental heterogeneity creates opportunities for different species, each finding pools where conditions match its particular tolerances. The result is that adjacent pools, separated by mere meters, can host quite different communities, each pool supporting the species best suited to its particular combination of characteristics. This variation means that no two tide pools are exactly alike, each one unique in its configuration of depth, exposure, substrate type, and resident species. Some pools have sandy bottoms where organisms can burrow. Others are carved entirely from rock with no sediment at all. Some contain large boulders that create sheltered microhabitats. Others are smooth bowls with little structural complexity. The physical character of each pool influences which species can thrive there, creating patterns of diversity that reflect both biological and physical factors. A naturalist visiting tide pools learns to read these patterns, predicting what species might inhabit a pool based on its physical characteristics before even looking closely. While physical factors shape communities, biological interactions determine the fine details of who lives where. Competition for space can be intense, particularly on rocks suitable for attachment. Barnacles compete with mussels, both trying to occupy the same surfaces. Fast-growing algae can overgrow slower species, shading them and monopolizing light. Mobile animals compete for the best hiding spots, the safest crevices where predators cannot reach. These competitive interactions sort species into patterns, each species occupying the locations where it competes most successfully, or where competition is least intense. The physical space of the tide pool becomes divided among residents through ongoing negotiation, boundaries constantly tested and occasionally redrawn. Predation overlays these competitive patterns with its own influence. Where predators are abundant, prey species may be eliminated entirely or restricted to refuges where predators cannot reach. The removal of a key predator can allow prey populations to explode, potentially leading to overgrazing or monopolization of resources. The balance between predation and competition determines much of tide pool community structure, these two forces pushing and pulling species distributions until a dynamic equilibrium emerges. This equilibrium is never permanent, always shifting as populations fluctuate and conditions change, but at any given moment it creates the patterns we observe when we examine a tide pool. Among the predators shaping these communities, sea stars have received particular attention from ecologists studying their keystone role. Research in Pacific Northwest tide pools demonstrated that removing sea stars led to mussel dominance and dramatic loss of species diversity, while pools with sea stars maintained diverse communities. This discovery helped establish the concept of keystone species, organisms whose impact on their ecosystem is disproportionately large relative to their abundance. Not all species are equally important. Some, like sea stars in certain tide pools, hold communities together through their ecological role. Understanding which species are keystones helps focus conservation efforts on protecting the organisms that matter most for ecosystem function. Not all keystone species are predators. Some are habitat creators, organisms whose physical structures provide essential resources for many other species. Mussel beds, as discussed, create habitat that supports dozens of associated species. Certain algae provide shelter and food for communities of small invertebrates. Even some mobile species like sea urchins can be keystones, their grazing maintaining the balance between algae and bare rock that many species require. The keystone concept has evolved beyond its original predator-focused definition to recognize that many types of organisms can play disproportionately important ecological roles. While ecologists study these patterns, the organisms themselves simply live, unaware of concepts like keystones or competition. A barnacle filtering plankton from the water does not know it is competing with the mussel beside it. The sea star pulling open a clam does not understand its role in maintaining community diversity. Each organism responds to immediate stimuli: M9, food, danger, mates, according to instincts refined by evolution. The ecological patterns we observe emerge from these individual behaviors, scaled up across populations and integrated over time. The barnacle lives its small life, and somehow, aggregated with millions of other small lives, creates the phenomenon we call an ecosystem. This gap between individual experience and ecosystem pattern is worth contemplating. Each organism in the tide pool has some form of subjective experience, however simple. The limpet scraping algae experiences something, even if that experience is utterly alien to human consciousness. The crab scuttling between rocks processes information about its world, makes decisions, acts on those decisions. These individual perspectives are real and immediate, while the ecological patterns we study are abstractions, human interpretations of aggregated effects. Both levels are real, but they exist in different ways. The organism's experience is immediate and felt, while the ecological pattern is observed and analyzed. This duality extends to our own relationship with tide pools. We can study them scientifically, measuring species abundances and interaction strengths, testing hypotheses about how communities assemble. We can also experience them aesthetically, appreciating their beauty and complexity without analysis. Both approaches are valid and complementary. The scientist and the poet see different aspects of the same system. Neither view complete alone. The tide pool invites both careful measurement and quiet wonder, rewarding those who bring clipboards and those who bring only open attention. Among the wonders that tide pools offer, bioluminescence appears in some species, creating moments of magic when disturbance triggers light production. Certain dinoflagellates living in tide pool water emit blue-green light when jostled, and on dark nights a hand moved through the water can leave a trail of sparkles. Some small crustaceans produce luminescence, as do certain species of worms and jellyfish that occasionally appear in pools. This cold light, produced through chemical reactions that convert chemical energy directly to photons, serves various functions: startling predators, attracting mates, communicating with others of the same species. For human observers, bioluminescence seems almost supernatural, life creating light through no flame or electricity, pure biochemistry transformed into visible energy. The chemistry underlying bioluminescence involves molecules called luciferins that react with oxygen in the presence of enzymes called luciferases. The reaction produces photons of specific wavelengths, determined by the particular luciferin-luciferase combination. Different organisms have evolved different bioluminescent systems independently, arriving at similar solutions through separate evolutionary paths. The widespread occurrence of bioluminescence in marine organisms suggests that producing light provides significant advantages despite the metabolic costs involved. In the competition for survival, the ability to create light has proven valuable enough to evolve repeatedly. Beyond bioluminescence, tide pools display other optical phenomena that catch the eye. Water in pools can be remarkably clear, allowing visibility to the bottom even in pools several feet deep. The water surface acts as a window, refracting light and sometimes creating lensing effects that magnify objects on the bottom. On calm days, the pool surface becomes a mirror, reflecting sky and clouds, the boundary between water and air nearly invisible. Light plays through moving water in patterns called caustics, concentrating and spreading as wavelets pass, creating dancing patterns of bright and shadow on the pool bottom. These optical effects transform tide pools into natural art installations. Light and water and life, collaborating to create beauty. The colors in tide pools range from subtle earth tones to brilliant hues that seem impossible in nature. Pink coralline algae encrust rocks in patches that look painted. Purple and orange sea stars provide bold contrast against gray stone. Green anemones cluster like impossible flowers. Red and gold seaweeds wave in gentle currents. These colors serve biological functions: camouflage, warning signals, attracting mates, but they also create compositions that human eyes find beautiful. There is no reason the biological functions and aesthetic appeal should align, yet they do. suggesting perhaps that what we find beautiful reflects deep patterns in how living systems organize themselves. This beauty draws people to tide pools, making them among the most visited and loved of all coastal habitats. Families come to explore, children discovering crabs and sea stars with delight. Students visit for biology classes, learning ecology through direct observation. Artists and photographers seek inspiration in the patterns and colors. The accessibility of tide pools, requiring no boats or special equipment, just willingness to walk rocky shores and look carefully, makes them democratic wonders, available to anyone who can reach the coast. They are nature's aquariums, free and open, maintained by tides rather than technology. Yet this accessibility makes tide pools vulnerable to overuse. Popular pools can be visited by hundreds or thousands of people each year, each visitor potentially disturbing organisms or removing specimens. Educational programs increasingly emphasize tide pool etiquette, looking without touching, replacing overturned rocks, avoiding stepping on organisms, leaving everything as you found it. When practiced, this careful stewardship allows tide pools to remain healthy despite high visitation. The challenge is balancing access with protection, allowing people to experience these ecosystems while minimizing impact. Some areas have succeeded through education, interpretation, and sometimes regulation, creating models for sustainable tide pool tourism. The educational value of tide pools extends beyond simple species identification. They teach ecological concepts like food webs, adaptation, competition, and succession, in contexts where these abstractions become concrete. A student watching a sea star hunt a mussel sees predation happening in real time, no longer just a textbook concept. Observing barnacles packed tightly on a rock makes competition for space tangible. Noticing zonation patterns, different species at different tidal heights, illustrates how environmental gradients structure communities. These lessons stay with learners, grounding ecological understanding in lived experience rather than abstract theory. Beyond formal education, tide pools foster scientific literacy and environmental awareness in broader populations. People who spend time observing tide pools often develop deeper appreciation for marine ecosystems and conservation. They see complexity and beauty in small places, learning that nature need not be exotic or remote to be fascinating. This connection between experience and conservation ethic is difficult to quantify, but clearly important. Protected areas typically enjoy stronger public support in places where people have personal experience with the ecosystems being protected. The tide pool becomes an ambassador for the ocean, representing marine life to people who may never visit offshore waters. While humans visit tide pools briefly, some organisms spend their entire lives within these small worlds. For a limpet or barnacle born in a tide pool, that pool is the entire universe, everything that exists. The limpet does not know about oceans beyond its pool, about continents and planets and stars. Its world is rock and water, algae and neighbors, the daily rhythm of tides. This limited perspective is not a lack, but simply the reality of being a limpet, an existence complete within its own bounds. The pool is not too small for the limpet. It is exactly right, containing everything the limpet needs. This thought leads to questions about scale and perspective. What seems tiny and confined to us is immense to smaller organisms. The amphipod living among algae fronds experiences that small tuft as a forest, complex three-dimensional habitat offering food and shelter. The copepod drifting in the water column knows that water is an ocean, vast relative to its own size. Each organism inhabits a world scaled to its own dimensions, and at every scale there is complexity, challenge, opportunity. The tide pool is not one world, but many nested worlds, each experienced differently, depending on the size and nature of the observer. These nested perspectives remind us that reality is not singular, but multiple, each organism constructing its own version based on its sensory capabilities and cognitive processing. A crab sees ultraviolet light that we cannot perceive. An anemone senses chemicals we would never notice. A fish detects vibrations through lateral line organs we lack. Each species samples different aspects of the environment, building different models of what exists. The tide pool contains all these versions of reality simultaneously. Objective features filtered through subjective perceptions to create experiential worlds that overlap but never fully coincide. While organisms construct their individual realities, the tide pool continues through time, outlasting its individual inhabitants. A barnacle may live a year, a sea star 5 years, an anemone perhaps 20. But the pool itself persists for decades or centuries, until geological processes finally erase it. Countless generations of organisms live and die in the same pool, each experiencing it anew, none aware of the deep history. If the rock could remember, it would hold memories of thousands of lives, the accumulated experience of every creature that ever sheltered in its depressions. But rock does not remember, and each generation begins fresh, discovering the tide pool as if for the first time. This amnesia of generations means that ecological knowledge cannot accumulate across time in the way human knowledge does. Each barnacle must learn for itself what its ancestors already learned: where to settle, how to feed, how to survive exposure. Evolution builds general solutions into genetics, instincts and physiological adaptations that work across generations. But specific local knowledge dies with each individual. There is no oral tradition among limpets, no written history of tide pools. Each present moment is all there is, experience ending at death rather than accumulating into cultural knowledge. Yet in another sense, knowledge does accumulate, encoded not in memory, but in genes. The adaptations we see in tide pool organisms represent millions of years of accumulated evolutionary wisdom. Each trait has been tested across countless individuals and countless generations, refined through survival and reproduction, optimized through the pitiless efficiency of natural selection. The limpet's shell shape, the anemone's stinging cells, the crab's behavior, all represent solutions to problems, answers to questions posed by the environment. This genetic knowledge is distributed across species, each lineage carrying its own hard-won insights, the collective result forming the adapted community we observe. This evolutionary perspective makes clear that tide pools are ancient habitats, likely existing since the first oceans formed billions of years ago. The earliest marine life may have originated in tide pools or similar marginal environments where chemicals concentrated and reactions could proceed. Throughout life's history, intertidal zones have provided habitat for countless species, arenas where evolution explored possibilities. The specific organisms have changed. The trilobites and ammonites are gone, replaced by crabs and snails. But the fundamental challenges remain: exposure to air, temperature fluctuation, salinity variation, kame. These problems have existed since the beginning, and life has repeatedly found solutions. The continuity of tide pools across deep time creates a kind of conversation between past and present. The organisms you observe today descend directly from organisms facing similar challenges millions of years ago. The solutions they employ, camouflage, burrowing, tolerance, resistance, are refined versions of solutions their ancestors pioneered. When you watch a limpet grazing on algae, you witness behavior that has occurred in intertidal zones since the Cambrian period. 500 million years of limpets scraping rocks by the shore. This connection to deep history makes even ordinary behaviors feel sacred, each moment linked to vast spans of time. While the past stretches behind us, the future remains unwritten. Tide pools will continue as long as there are rocky coasts and tides. But which species will inhabit them depends on choices we make now. Climate change, ocean acidification, pollution, and overharvesting all threaten tide pool communities. Yet these threats are not inevitable. They result from human choices and can be modified by different choices. The future of tide pools lies partly in our hands, in whether we value them enough to protect them, to make the changes necessary to preserve functional marine ecosystems. This responsibility can feel overwhelming, but it can also inspire action. Each choice to reduce carbon emissions, to protect coastal habitat, to minimize pollution, to practice careful stewardship when visiting tide pools, these choices matter. Aggregated across millions of people, individual actions create the collective impact that will determine which species persist and which disappear. The tide pool asks nothing of us, requires nothing, simply continues existing and adapting as it has for billions of years. But we, having the capacity to help or harm, bear some ethical weight. We can choose to be tide pool guardians rather than tide pool destroyers. Among the reasons to protect tide pools is their role as indicators of ocean health. Because they concentrate oceanographic conditions and biological communities into small, observable spaces, tide pools can reveal broader patterns in marine ecosystems. Changes in tide pool communities often signal changes in offshore environments. Scientists monitor tide pool organisms as sentinels, early warning systems for larger problems. A decrease in sensitive species, a shift in community composition, changes in reproduction or growth rates. These signals from tide pools help us understand how human activities affect the ocean. The pools become diagnostic tools, their clarity making visible processes that would be difficult to detect in open water. This monitoring role makes tide pools valuable beyond their intrinsic worth. They are research sites, natural laboratories, teaching tools, and indicators. They provide ecosystem services, shoreline stabilization, nutrient cycling, food production for offshore species. They support fisheries by serving as nurseries for commercially important fish. They protect coastlines by absorbing wave energy. These utilitarian values may motivate protection efforts, even among those who do not value nature for its own sake. The argument for tide pool conservation can be made economically, scientifically, and ethically, appealing to different values and perspectives. Yet perhaps the strongest argument for protecting tide pools is simply that they are wonderful, that they add beauty and interest to the world, that knowing they exist and thrive makes life richer. There is value in diversity, in complexity, in the knowledge that strange and marvelous creatures pursue their lives in small pools by the shore. This aesthetic and spiritual value defies quantification, but feels real to those who experience it. Standing beside a tide pool and watching life's intricate dance creates moments of connection and wonder that remind us we are part of something larger, embedded in living systems that preceded us and will continue after we are gone. These moments of connection are gifts that tide pools offer freely. They ask no admission, require no reservation, simply exist in their twice-daily rhythm, waiting for anyone who cares to look. The accessibility and generosity of tide pools make them democratic treasures. Natural wonders, available to all. In a world where much natural beauty is remote or restricted, tide pools remain open, inviting us to crouch beside them and peer into their depths. They are patient teachers, revealing their lessons to those who take time to observe, reflecting back to us the complexity and resilience of life. And now the tide pools fade behind your thoughts. We have drifted through 100 quiet truths about these small worlds, caught between land and sea. From the twice-daily rhythm of tides, to the organisms that endure exposure, from barnacles filtering plankton to sea stars prying open mussels, from the fierce competition for space, to the remarkable adaptations that allow life to flourish in conditions that shift with every tide. You are connected to this world. The same ocean that fills tide pools covers most of Earth's surface, touches every continent, connects all shores. The salt water in those pools contains the same minerals that flow in your blood. Legacy of our shared marine ancestry. Reminder that all life began in water and carries that beginning forward. The carbon and nitrogen and phosphorus cycling through tide pool food webs are the same elements cycling through your own body, temporarily organized into the pattern we call life, before returning to the environment to be organized again. Tonight, as you rest, tide pools around the world continue their ancient rhythms. The tide rises and falls, organisms hunt and hide, larvae settle and colonies grow. Waves wash over rocks, bringing fresh nutrients and carrying away waste. Sea anemones extend their tentacles into the current, patient as flowers that will never wilt. Limpets scrape their slow paths across stone, leaving trails that fade like memories. And you are safe, warm, resting in a world full of wonder that reveals itself to those who pause and look closely. If you are still awake, go ahead and watch the next chapter. Sleep well and dream softly of small pools holding oceans, of creatures that endure, of rhythms older than memory, of life persisting in the spaces between. Good night.